For this video, we will be focusing on the usage of BLAST, or the Basic Local Alignment Search Tool. BLAST is an algorithm for comparing primary biological sequence information, such as the amino acid sequences of different proteins or the nucleotides of DNA sequences. The BLAST program was designed by Stephen Altschul, Warren Gish, Webb Miller, Eugene Myers, and David J. Lipman at the National Institutes of Health. A BLAST search enables a researcher to compare a query sequence with a library or database of sequences and to identify library sequences that resemble the query sequence above a certain threshold. From the BLAST search also, different types of BLASTs are available according to the query sequences. For example, following the discovery of a previously unknown gene in the mouse, a scientist will typically perform a BLAST search of the human genome to see if humans carry a similar gene. BLAST will identify sequences in the human genome that resemble the mouse gene based on similarity of sequence. These are the steps for searching a BLAST. First, choose a desired protein BLAST. Secondly, enter query sequence. Next, choose non-redundant database. Then tick on the SciBLAST for algorithm. And lastly, click on BLAST to proceed with the results find. From the search, you will receive a conserved domain and the BLAST of the desired protein. First, we will look on the conserved domain. Conserved domains contain conserved sequence patterns or motifs, which allow for their detection in polypeptide sequences. This is the conserved domain of 2HT5A, that is, obtained from the BLAST search. And this figure shows the BLAST result of 2HT5A, that is also retrieved from the search. From the BLAST, we will now proceed with the usage of InterProScan. So, basically, what is InterPro? InterPro is a resource that provides functional analysis of protein sequences by classifying them into families and predicting the presence of domains and important sites. To classify proteins in this way, InterPro uses predictive models known as signatures, provided by several different databases that are referred to as member databases that make up the InterPro consortium. Meanwhile, InterProScan is the software package that allows sequences to be scanned against InterPro signatures. InterPro is useful because it combines signatures from multiple, diverse databases into a single searchable resource, reducing redundancy and helping users to interpret their sequence analysis results. By uniting the member databases, InterPro capitalizes on their individual strengths, producing a powerful diagnostic tool and integrated resource. InterPro also provides functional analysis of proteins by classifying them into families and predicting domains and important sites. This will produce a powerful integrated database and diagnostic tool. Therefore, for our group, this is the result for 2HT5A. As you can see, there are the family result of 2HT5A and the domain for it. This is the family description of the 2HT5A received upon clicking on the previous image. As described in the result, the family for 2HT5A is the glycoside hydrolase, family 34. This is the domain description, also obtained from the InterPro. The domain for 2HT5A is sialidosis. Next, we will be using the Catalytic Site Atlas, or CSA, The Catalytic Site Atlas is a database documenting enzyme active sites and catalytic residues in enzymes of 3D structure. We defined a classification of catalytic residues which includes only those residues thought to be directly involved in some aspect of the reaction catalyzed by an enzyme. The C.S.A contains two types of entry. Number 1 is the original hand-annotated entries derived from the primary literature. References for these entries are given. The second one is homologous entries found by sequence comparison methods to one of the original entries. The equivalent residues, which align in sequence to the catalytic residues found in the original entries, are documented. These are the steps to obtain C.S.A data. The catalytic site for our protein of interest, which is 2-HT5A, was not annotated. Therefore, we proceed with the nearest family of 2-HT5A by entering its EC number, which is obtained from the family description of 2-HT5A. And this is the result of our findings. Multiple sequence alignment, or MSA. MSA is generally the alignment T of 3 or more biological sequences, protein or nucleic acid, of similar length. From the output, homology can be inferred, and the evolutionary relationships between the sequences can be studied. From the resulting MSA sequence, homology can be inferred, and phylogenetic analysis can be conducted to assess the sequence's shared evolutionary origins. So, these are the steps in obtaining a MSA. As mentioned earlier, the MSA would help in obtaining evolutionary relationships between sequences. Thus, a phylogenetic tree is obtained from the MSA. A phylogenetic tree, or evolutionary tree, is a branching diagram, or tree showing the inferred evolutionary relationships among various biological species or other entities. The branch lengths indicate the amount of character change between the species. These are the steps to obtain a phylogenetic tree. This is the tree obtained from our group MSA. As you can see, the shorter distance indicate a higher similarity of structure can be seen between 2-HT5 and 3-Beaker. However, greater evolution occurs between 2-HT5 and 4-HZV since they are more distant from each other. The 1V0Z proteins may belong to a higher group since the branching is much earlier than others. Lastly is the .ln file. The file can be read only using Microsoft Word software. Therefore, the alignment of sequences can easily be shown, and these are the results.